Jamie, it kind of looked like your cursor was moving around on there.
Yeah, because I'm sharing that screen, so that's where my stuff is to control it. So.
Well, can Barbara do that?
It's just when she shares her screen, I don't know what's happening. Maybe in this mode of work, we were in a practice mode before, so.
Maybe.
You were just using your cursor to share?
Yeah, see, can you see it?
No, I can't see it.
It's a little hand.
It's on her face.
Oh, yeah, I just did. Yeah, okay. Yeah, well, we'll see. Maybe I can use that. But if not, it is what it is.
You just have to use your words to be descriptive.
Exactly.
Okay.
Good afternoon, everyone, and thank you for joining us for our first ever 2020-21 New Frontiers in Biomedical Research Seminar Series. I am Dr. Mary Calderera Moore, and I'm an Associate Professor in Biomedical Engineering, and I'm joined here today with my colleague and series co-organizer, Dr. Jamie Newman, who's an Associate Professor of Biology in the School of Biological Sciences. As we began this school year in the midst of the health crisis none of us had ever experienced, we thought it was important that we continue the series to address ongoing research and discuss the challenges and solutions we continue to face within the pandemic associated with SARS-CoV2 and COVID-19. Unlike our previous years, we have developed a theme for the series in which we are going to take the opportunity to dive deeper into research and impacts and outcomes associated with COVID-19 pandemic, and also things broadly about science and medicine with the series on the biology of the disease, which will be the focus of the fall quarter, patient care and healthcare, which will be the focus of the winter quarter, and finally, the role of scientific communication in higher education as we go forward. We appreciate you for taking the time out of your day to join us and hope you will enjoy our first presentation. Dr. Jamie Newman is now going to introduce our guest and panelists for the day.
Yeah, thank you, Dr. Calderera-Moore. It is now my great pleasure to introduce our guest for today. Dr. Barbara Osborne is Distinguished Professor at UMass Amherst in the Department of Veterinary and Animal Sciences. She earned her PhD in genetics at Stanford University and went on to complete her postdoctoral training at the NIH. Dr. Osborne's research interests have focused on various aspects of immune function. Her early studies in immunoglobulin gene diversification identified gene conversion as a novel mechanism of diversification in murine immunoglobulin genes. In the early 1990s, her research focus shifted to understanding how T lymphocytes die. Her lab was the first to identify and isolate genes that regulate apoptosis in mouse T cells. Over the years, her laboratory has trained numerous graduate students and postdoctoral fellows. We are also joined today by Dr. Gordon Love, a professor and chair of the Department of Pathology at Louisiana State University School of Medicine and the medical director of laboratories for University Medical Center, both in New Orleans. Dr. Love graduated from Louisiana Tech University with a BS in mathematics in 1973 and from Tulane University School of Medicine with an MD in 1978. During his career, Dr. Love directed several hospital-based laboratories and worked for Quest Diagnostics as Western Regional Medical Director in charge of West Coast operations. While in California, he was Professor of Pathology with University of California Davis School of Medicine. Dr. Love has published a number of articles in pathology journals and authored 2 books in the field of fungal diseases. Finally, I am joined by Dr. Pat Hindmarsh, an associate professor in the School of Biological Sciences at Louisiana Tech, who will join me in moderating the discussion that will follow our 2 short presentations. Please feel free to submit questions through the Q&A window, and we look forward to answering as many of them as we can. Thank you again for joining us, and I will now turn it over to Dr. Osborne.
Minutes upload. Okay, folks, can you see my screen? Shake your head. Yep, good. Okay. Thank you very much, Jamie, Mary, Pat, for inviting me. I'm really excited to be here and to tell you something about the immune response to COVID. So what I want to first do is talk about where the immune system lives and how it gets activated, and then we'll talk about how it responds to COVID. So I don't think you can see my pointer. In fact, I know you can't see my pointer. But if you look on the left-hand side of this figure on your screen, you'll see a series of blue lines and blue dots that represent the lymphatic system and the lymph nodes. You'll also see over on the right-hand side a blue spleen living in the middle of this figure. This is where the lymphoid system lives, and this is where it mounts an immune response to pathogens. In yellow, you will see the bone marrow and the thymus, and this is where the lymphoid system develops. But once it develops, it matures, it moves out into these blue areas of the lymphatics, the lymph nodes, and the spleen. And let me see if I can advance this for you. Okay, so let's go over very briefly. This is a complicated slide, and unfortunately my pointer's not working for me, so you're going to have to bear with me as I take you through this slide a little bit fast and you can just ignore parts of it. But if you look on the left-hand side of the slide, you'll see where I have pathogens infecting cells. Once the pathogen infects the cell, you get a response that we call the innate immune response. This occurs very rapidly within a few minutes to hours after a pathogen invades your body. And if you look just to the right of those infected cells, you'll see it's a green cell called a monocyte. Well, that monocyte or macrophage becomes activated and becomes an activated macrophage, and it secretes a number of immune cell hormones that we call cytokines. Cytokines are merely the hormones of the immune system, and they mediate immune responses. So that activated macrophage, once it gets activated, makes all of these cytokines, which induces and drives what we call the adaptive immune system, which is on the right-hand side of the slide. And the adaptive immune system is unlike the innate immune system, which is nonspecific. It makes any pathogen will induce these cytokines. But the adaptive immune system will create a response that is specific for that pathogen. So it will only respond to SARS-CoV2, will only respond to influenza. It's very antigen or pathogen specific. And this is mediated by the white blood cells we know as lymphocytes, and in particular T cells and B cells. So the activated macrophage makes the cytokines which activate CD4 and CD8 T cells. And in the context of today's talk, we're going to talk about CD4 T cells because those T cells are become a subpopulation we call T helper cells. And T helper cells actually help B cells make antibody. And it's these antibodies that are going to be very important in clearing infection by COVID. So, see if we can go down. Let's talk very briefly about antibodies and how they're made. I want you really to concentrate on underneath where it says the peripheral lymphoid tissue. So this is where B cells are developing to become antigen-specific B cells. So during development in the bone marrow, B cells arise that have different antibodies on their cell surface, and we're not going to go through exactly how that occurs because it can take me 2 lectures normally to do that. But for today's talk, just assume that these B cells each have a different antibody on their cell surface. When the B cell sees an antigen that is recognized by that particular antibody, that B cell that you see here in the middle of the slide near antigen 2 is expanded and rapidly proliferates. And those cells proliferate until you have hundreds, thousands, millions of those B cells that have then differentiated into plasma cells, which are really the end stage of B cell differentiation, and they make millions and millions of antibodies. Antibody production by these B cells can produce up to 10 milligrams per mil of antibody in your blood. So if that antigen is COVID-2, these B cells can be all making anti-COVID-2 antibody. Also important to note is this process of maturation and development of an immune response develops what we call memory cells. And let's talk for a moment about memory. So if we go down here on this figure and you look up to the origin of this figure on the x-axis, you see the primary response to antigen. And the primary response to antigen, there's a lag period of several days, maybe even a week or 2. So after you see antigen, the pathogen, it takes a while for the B cells to start making antibody. And they make a relatively low level of what we call IgM antibody, usually a lower affinity antibody than IgG. Eventually, these B cells start to make IgG antibody, but it's still a pretty low level. And this is called the primary response. If you see antigen again, for example, if you're immunized, and you have received your first dose of immunization, and now you receive your secondary dose of immunization here in the center of the figure, you see that there's no lag phase. That response is rapid, and it's couple of log orders higher amount of antibody that's produced. And additionally, this antibody is IgG. And so the secondary response that we see here is driven by those memory B cells that I mentioned on the previous slide. So now let's talk, let's see if I can get to it. Let's talk about how COV2 infects cells and initiates this response. So what you have here on the left-hand side is SARS-CoV2, and it binds to the ACE2 receptor that's found in the ciliated epithelium of the lung. And once it binds to the receptor, it injects its RNA into the cell, and that draws in, that actually initiates the production of cytokines, which draw in alveolar macrophages that become activated, release cytokine, and activate the adaptive immune response. So it's this interaction that will activate the adaptive immune response. So what can happen in some cases, though, however, these alveolar macrophages can get overactivated, produce a lot of cytokines, which we call a cytokine storm. And it's this cytokine storm that can actually make people very sick. And I'm sure Dr. Love will talk to you about the pathology of what happens after a cytokine storm. All right, so if, however, you have a good antibody, if you've been immunized to SARS-CoV-2 or you've seen it previously, you will make an antibody response, just as we talked about a few minutes ago. And the good thing about these antibodies is they will bind to this, especially if you make antibodies to the spike protein, which is actually the coat of the SARS-CoV-2 virus. The antibodies will bind to spike protein, and this antibody binding to the spike protein will prevent the virus from binding to its receptor. So the cells in the lung do not get infected, you don't make the cytokine storm, and you actually prevent the infection from taking place. The immune system will clear these antigen-antibody complexes and will kill the virus. So I also want to introduce the concept of immunization by a virus infection or immunization versus vaccination. So what happens if you become infected by the virus is you get infected by the virus, you make an antibody response after the infection, and that's the red line, and that's our primary response. However, if you have been vaccinated, you've already made an antibody response, and which will diminish over time. But then if you see the virus, as you see here on the left-hand side, or excuse me, the right-hand side of the slide, you have those memory cells which will give you a response very, very rapidly. So on the left-hand side, where you have the virus initiating the antibody response, you get an antibody response, but it's after you've already been infected. However, with vaccination, you get this happening very, very quickly, and that's to your advantage, obviously. Do I have a few more minutes, Jamie?
Sure.
Okay, so I just want to very, very quickly mention that we have ways of testing for infection as well. And what we can do, and you've all heard of these, there are three major ways that people are using right now in the clinic to test for whether you have become infected. We have over here on the right-hand side the PCR test, which you've all heard about, which is very, very highly specific, and it's very, very accurate. However, it requires a lot. It requires labor. It's labor intensive. It requires a lot of reagents and specialized equipment. On the left-hand side, there are rapid serology antibody tests, which you can actually do at home, but they're very low in cost, but they also have a low degree of, well, let's put it this way, they're not as accurate as the PCR test. And I also just want to quickly mention that there is a new test that's been developed at Yale Medical School called Saliva Direct, where you actually can collect saliva, people can spit into a tube, and that tube also is analyzed by PCR. But this test is very rapid. It's rapid. It takes a couple of hours, but it must be done in a CLIA certified facility. However, it is easy sample collection and uses easily available reagents. So I think that this is going to be one of the ways we are testing in the future, because it's much more efficacious, it's much cheaper, and it's much easier to do than the time-consuming PCR analysis we do currently. And I just want to end up by saying that the immune response to COV2 declines with age. So people who are young, many of you who are listening to this seminar, you'll mount an antibody response and a T helper and a cytotoxic T cell response, which we didn't talk about, all of which will kill the SARS-2 virus. However, as you get older and you're my age, you may only initiate one of these responses, maybe only antibodies or T helpers or CTL responses, and you won't respond as well to SARS-2. And that may be the reason, in this paper that just actually came out a couple of days ago in Cell, that may be the reason why older people are more susceptible to the virus. And there's one last slide that I think it's important to share with you, and that is to mention that SARS-CoV-2 has a lower fatality rate, but a much higher rate of transmission than its cousins, SARS-1 and MERS. And so as you can see, we have a lot more cases of SARS-CoV-2, and that's because it has a much higher rate of transmission, but luckily it is not as fatal as MERS or SARS-1. And what people are really concerned about, we will have another SARS. There'll be SARS-CoV-3, SARS-CoV-4. It's just a matter of time. And what we really worry about, if SARS-CoV-3 or SARS-CoV-4 combines the transmissibility rate of CoV-2 with the fatality of MERS, for example, we're in for big trouble. So the argument I want to make is we need to be ready for these other viruses, not only SARS, but other viruses that are going to arise. And we need to be prepared, and which we aren't right now. We need to be prepared for these and get our ducks in a row, so when it happens, we are ready and we can fight it much more quickly than we have in the past. So we need to have viral memory, just like we need immunological memory. And I'm going to end there.
Thank you, Barbara. I think that's a good way to segue to Dr. Love, who will tell us more about pathology. And at the end, we will have the opportunity for questions and discussion specifically related to the presentation, but also broadly. I plan to ask both our speakers how we prepare for that next infection in our future, and what we've learned from this current pandemic. So with that, I'll introduce Dr. Love, and it's all yours.
Okay, can everyone hear me okay? Oh.
Gordon, we lost your slides.
Can you hear me okay?
Yeah, your slides were there. You want to try that again?
Okay, I'll start again.
Okay.
Can you see?
Yep.
Okay, good. No, I enjoyed Dr. Osborne's presentation. Mine is going to be a little bit different. I'm going to go through our autopsy findings here at University Medical Center and LSU through this epidemic. I'm going to first kind of tell you the role that I work in, the facility here, and I'm going to go a little bit and tell you a little bit about what a pathologist is, because not everyone understands that. Then I'm going to go through our autopsy findings that I think have an important role in trying to understand what we're dealing with in the human disease caused by this SARS-CoV-2. I work at the University Medical Center. It's a 3-year-old facility, $1.2 billion of taxpayer money. Very much enjoy working here. Another view, basically there's 3 bed towers, and there's like an E. There's a support facilities and laboratory are in back. The hospital, this is about a year or so old. You can kind of get the idea. And now we have 7 hospitals, much greater than 1 million patient population, probably closer to 1.6 million billable tests annually in the laboratory, closer to 2,500 specimens received on our automation line daily. I just wanted to touch bases to show you that, and since you're biomedical engineers, I thought you'd be interested. We have one of the largest automation lines in clinical chemistry in the country. It's a U-shape. Basically, the inlet is over here. There's centrifuges and decappers. Our infectious disease testing is here. Our mainline chemistry analyzer is here. We have our hematology and coagulation instruments here, and these are storage areas where the tubes can be stored for 3 to 4 days. If follow-up testing is needed, they can be withdrawn and stored and tested without the need for any human intervention. About 3 or 4 medical technologists can run this whole thing. Just quickly to show you a photograph, I enjoy walking through this every day. This is the exit area. We're heavily veterinary culture and instrumentation. It does a great job. So I showed you where I work, what we do, and what is a pathologist? I just spent a couple moments talking about it. A scientist who studies the causes and effects of disease, especially one who examines laboratory samples of body tissue or fluid for diagnostic or forensic purposes, including both tissue evaluation for tumor and fluid chemistry evaluations. A number of anatomic pathology specialties in pathology, and a number of clinical pathology specialties. I'm a microbiologist, I'm also a chemist, and I'm a cytopathologist. A lot of people think that pathologists just do autopsies, and that's false. So we do a lot of things, but autopsies certainly were very important in dealing with this outbreak. Now I want to take you on a journey of how we progressed and how we dealt with the coronavirus outbreak. Here's a listing of our cases. There's patients on ventilation, in the ICU not ventilated, in acute care or ED. This is 19 March. We got our first patient at University Medical Center on the 10th of March, here somewhere. And we had kind of an exponential rise in the number of cases. Now, at first we had no testing available, and I was able to set up testing with Tulane on the 30th of March, which was just huge fast for getting this going. We instituted testing at the University Medical Center on April the 13th. We brought in 2 more platforms on April the 20th. This is just huge fast for instituting new testing. Now, having let you know what's a pathologist, what I do, where I work, now let me take you through the journey of how we approach autopsies and how I think that this effort was important. The first thing I did in February, as it was clear that this was a locomotive coming down the line, it's going to hit us soon, is I went to our morgue and I confirmed its operation with the facility engineer. And the CDC recommends to do COVID autopsies that you have a negative pressure room, that you have minimum of 6 air exchanges per hour, and that the exhaust is exhausted directly to the outside through a high efficiency particulate aerosol filter, HEPA filter. Since we had a recently built facility, we had all of these features. Now, many facilities and morgues in the United States did not. So it turned out that we were one of the few places in the United States that actually did autopsies. Now, I want to show you this photograph. This shows our morgue. This is me, and you can see the guy here. But I wanted to tell you, this is a bunny suit. And I'll bring it up a little bit later, but I wanted to see what a bunny suit looks like. Now, it doesn't have ears. It has everything else. You put it on, you zip it up, your hands come out, and your face is out. But otherwise, you're completely enclosed in an impervious covering. And again, this is our morgue. Here's this is not a COVID autopsy, but just to show you a photograph of our morgue. Now, let's talk about, then I examined CDC recommendations for personal protective equipment for COVID-19 autopsies. The CDC recommended basically 2 layers of clothing, the impermeable isolation gown and an apron with a face shield and an N95 respirator. We weren't too impressed with this. Now, one of our this is one of our 4th-year pathology residents, Bing Han, and he contacted an infectious disease colleague in Wuhan City for PPE recommendations. Now, basically in China, they had determined that 3 layers of body coverings were helpful, and goggles were necessary for eye protection. So we implemented this, basically a bunny suit, an apron, and impermeable surgical gown. So this is the result, and this is before we did any COVID autopsy. Basically, this individual has a bunny suit. You can see the hood here. Now, this basically covers her legs and everything. Over this, she has an apron, which you can't see, and over this, you have a surgical gown. You have layers of gloves. She has goggles, she has an N95 respirator, and a face shield. And she has a boot over her, over the bunny suit foot attachments. So we decided that this provided complete protection, or as good a protection as we could do for people doing COVID autopsies. So we had both the facility, we worked out the PPE. And I might quickly say that this was written up in College of American Pathologists Today, which is a periodical. It came out in May, where I discussed this. I want to say this is Bing Han. This is Dr. Vanderhyde, who wrote the first couple articles from our facility, and I'll show you one of those in a moment. He's a cardiac pathologist in our department, and he's the head of the morgue. This is Dr. Jack Herbert, who I felt very grateful to him. He said he felt very, very safe going into this unknown experience. I want to say, too, that I put a photograph here. Dr. Crawford will be talking to you in a little while on testing. Basically, he was the head of Tulane, and he and I brought up the first testing on March or April the 30th, which is really fast processing. So we cooperated to bring up the first coronavirus testing in our joint facilities. This is the first, and also this was published in Archives of Pathology and Laboratory Medicine, where we went over both the facilities and the PPE, so other places, pathology departments, to see how to do it. Now let me go now into COVID-19 autopsies. And this is what Dr. Vanderhyde saw in his first autopsy. And you can see immediately that these are very abnormal lumps. It kind of looks almost like liver. But you can see what's very abnormal here is that this is a premortem clot, and the premortem clot is not as shiny as a postmortem clot. Here's another one, and this is a little out of focus. It's not shiny at all. There are a great many of these. Basically, all of the vessels are studded with these little dark clots. When we look at these under the microscope, these are capillaries, and they're just distended with these thrombi. There's a little higher power. Again, there's no red cells in these capillaries. There's another one here. We could stain these with CD61, which is a platelet marker, and these would be positive. You could also stain these with fibrin, and these were positive. So it's a platelet fibrin clot. And this is what killed these patients. And literally, we saw this be a systemic disease. Dr. Vanderhyde published this in Lancet Respiratory Medicine. This is one of the first articles going through the findings in COVID-19. Basically, he found thrombosis and microangiopathy in the small vessels and capillaries of the lung with associated hemorrhage that contributed to death. We found diffuse alveolar damage in hyaline membranes, and we found cardiac findings, including individual cell necrosis, which is very interesting. This is an unusual pattern. We published this separately in an article in Circulation. And I want to show you, this was picked up by the Washington Post in their July 1 issue, Coronavirus Autopsies, a story of 38 brains, 87 lungs, and 42 hearts. What was learned from the dead can help the living. This is an excerpt from the story. One of the first American investigations to be made public on April the 10th was out of New Orleans. The patient was a 44-year-old man who had been treated at LSU Health, which was studying along. We'll never forget that day. This is very unusual. We saw the same pattern. He was so alarmed that he shared the people online. The findings caused a stir in many hospitals, influenced some doctors to start giving blood thinners to all COVID-19 patients. This is the first recognition that the disease is causing an incredible blood clotting throughout the system. I will say that since then, the steroids has been the mainstay in treating these patients and has led to greater survival in the intensive care units. I think the blood thinners, I think there's an answer there. I don't think it's quite been worked out. It's a very odd clot that is platelet rich. It also includes fibrin. It's not the standard clot that you would see in disseminated intravascular coagulation. I want to go through some unpublished results just to give you a flavor of what we're working on. I developed a coronavirus histochemical stain. Basically, this uses a primary antibody. I obtained an antibody to the coronavirus nucleocapsid. It's picked up with a secondary antibody that's linked to an enzyme. A chromogenic substrate is applied, which stains the reaction, and you can see wherever the antigen is. And this is what it looks like in lung. Basically, the alveolar lining cells are lighting up very strongly here. Interestingly, we found that we've done about 34 cases of COVID-19. We found most of the lungs were completely negative for coronavirus by the stain. Now, the stain is somewhat has a higher level of detection than, say, PCR, but I think what happens is I think that the coronavirus starts the process and then drops out. And we found it's basically positive in the early part of the disease, and then we can't find the virus later. I carried this coronavirus in the immunohistochemical stain to venotubular cells, and they're strongly positive. These are glomeruli in the kidney. And as Dr. Osborne showed, basically the ACE2 is the means through which the organism gets into the cell. And ACE2 is rich in lung alveolar epithelial cells and enterocytes of the small intestine. It's also rich in venotubular lining cells. It's also found in most cells of the body. So I think what happens is you have a systemic spread of this virus, and it's found in greater amounts in some organs than others, but there's a very significant systemic spread based upon the presence of ACE2 receptors. And I'm getting close to finishing. The third thing that we haven't written up yet, we're working on it, is central nervous system disease. We didn't remove brains early on because it was considered that the bone dust was highly infectious, and a vacuum thaw, as this is, was recommended to remove the bone dust. I finally got mine in July, and I completed 5 autopsies, COVID autopsies at that time, and was able to remove a brain. I want to say as an aside, I was tempted to try to build this myself, but you have to draw the line somewhere, and I decided that buying one would be better. Our neuropathologist, Dr. Rose, has provided some very tantalizing photographs of central nervous system disease in COVID-19. Here's a neuron. You can see the processes, and there's an acute inflammatory attack. This is most interesting. This is a capillary. You can see a red blood cell here. There's endothelial cell proliferation here. And attached to this is a microabscess. You know it's an abscess that it contains PMNs. Dr. Rhodes has suggested that perhaps the virus breaks down the blood-brain barrier, which can lead to central nervous system infections. But this is there's a fascinating possibility here. This is other tidbits from Dr. Rhodes' evaluations. He's finding dilated tortuous microvasculature. There's in the temporal lobe and other areas. You can see almost tuft-like appearances. He's describing this as ampule-like shapes. That is, this is like the bottle, and there's a constriction here, and which he calls ampule-like. It's kind of an interesting term. Also, he's finding microthrombi and microvessels, like we saw in the lab. Basically, here's in the cerebellar cortex. You can see the thrombi easily here in the temporal lobe. You can easily see the thrombus here. These stain with CD61, as I mentioned, which is a platelet marker, but also they will stain with fibrin. In the last slide, he also found areas of lymphocytic proliferation. And we find these small nests in other areas in the lung and other areas that appears to be something of a marker of COVID-19. We can label these with different immunolabeling, CD3, CD8, and others, and we're doing this. So basically, we're progressing to try to understand the pathology of the disease. We're not a research organization per se, but we want to understand as much as we can about this disease, and this is where we've progressed so far. I'm finished.
All right, thank you, Dr. Love. So at this point in the seminar, we're opening it up to conversation and questions from the audience. Dr. Hindmarsh and I have a few to get us going, but please, if you have questions, feel free to drop them into the Q&A section, and we will ask them as we go. Actually, there was one quick question I guess we could start with from the audience. I guess at the end of your presentation, Dr. Osborne, you mentioned sort of this chain of we could have a CoV2, 3, 4. Do you know why CoV2 is named CoV2? Is it just the second of this class of virus that we've identified, or is there more behind that?
No, I'm not sure. I mean, there certainly are other coronaviruses, but I think maybe Dr. Love can step in and address this because he's a microbiologist. But I think SARS was the first of its class of coronaviruses, and COV2 was MERS was the second, and COV2 was the third. And I'm not sure how the nomenclature came into being.
If I may, can you? I have a graphic to show if you could sign.
Yeah.
Can I pick up or share?
Oh yeah, you bet.
Yeah.
Well, anyway, can you see the main thing?
Yes.
Basically, this shows the relatedness of the different known pathogen coronaviruses, and basically the relatedness to SARS-2. You can see SARS-CoV-1, which is here, is very closely related to the SARS-CoV-2. Actually, as they branch out the genetic tree, they're very closely related. So, and again, the SARS-CoV-1 had the outbreak in 2003, and there was a second outbreak in a Chinese laboratory, I believe, in 2004. And this is a very good thing for getting that under control. Now, the MERS is a little further out of line. So these are the cold viruses, the coronavirus cold viruses that circulate. And you can see that the relatedness here to the SARS-CoV2 is 65%, 55%, or less. So there is some significant relatedness, but these cold viruses tend to be lower affinity in the population. They don't cause that many colds, but they're different. There's a significant difference. And you can see that the MERS does differ from CoV1 and CoV2. I think the relatedness has to be very close, like 90 to 95%. So I agree. There'll be a CoV3, CoV4, and probably continue. And we may even see a MERS CoV2. So that's what I'm thinking.
That could be scary.
So to follow up on that, there was a question about predicting the sort of severity and fatality of another COVID disease-causing virus. Is it based on some of that pathology, maybe that it could be predictive, or is there any way to predict what might be our future?
I personally think it's hard to predict. I think we're dealing with bat viruses here, and what's going to come out of the bat next, I don't know. I tend to think that perhaps the incubator for this in China with the open meat markets and so on, with the different species being represented, that controlling that may be helpful to try to control the appearance of something new. But I don't know what's going to come out of that next.
Mm-hmm. Yeah, I guess I would also like to add, this is my plug as an experimental scientist, but I have a good friend who works in Chicago at Loyola University, and Tom Gallagher has been a guy who's worked on coronaviruses his whole academic career, and he has gone through these ups and downs in funding because for long periods of time, people don't really care about coronavirus. When SARS hit back in the early 2000s, yes, people cared about it. Then MERS, people cared about it again. Then they went for a long period of time where we didn't spend a whole lot of attention or money on coronaviruses. So my short answer is we should be funding people to do research in all sorts of pathogens, whether or not we're seeing a pandemic or epidemic or not. Because if we understand the biology of these viruses, we'll be able to help the physicians design the right approach to treating the patients.
Dr. Hindmarsh, if you could just ask any of your questions.
Yeah, Gordon, you'd mentioned that in that little tree that you had that showed the different coronaviruses and the relatedness of SARS-1 and SARS-2. But I was reading that there was some blood samples that were picked up several years ago that were being retested with the coronavirus or with SARS-2. And they were reacting to it, suggested there may have been some cross-reactivity between the normal common cold version and the present disease. And I was wondering, can we give people the common cold? Maybe get some herd immunity from that.
I think that's an inescapable idea. And it occurred to me that if somebody could offer me some of the low pathogenic potential coronaviruses, I might like having a cold. Because there may be significant cross-reaction.
Right. I mean, it appears to be some anyway. And it was just a very limited sample they were testing. So I think it's pretty intriguing.
I agree.
So there is a question from the audience about the effects on brain tissue. Yeah, have you seen any of the effects on brain tissue?
Yes, I have. I've stained our brain samples extensively with our coronavirus immunohistochemical stain. I've got no reaction whatsoever. Now, that doesn't mean that the virus isn't there. It probably definitely is there. I think just there has to be a threshold that is probably pretty significant for you to actually see it. I'm going to follow up with the in-situ hybridization coronavirus to see if that may be more sensitive to see if I can see virus. I think for sure there's the virus is being picked up by the endothelial cells. And I think that kind of fits into what we're seeing in terms of vascular proliferation and endothelial cell changes.
Another audience question. There are several vaccines in different trial phases and at different points along the process. If there were to be a vaccine that was well distributed, how long might it take for the COVID-19 virus to become less prominent? How long will it take for vaccines to actually work?
Well, I'll leave how long it takes to become effective for Dr. Osborne to answer. But I think that there would need to be a rather extensive compliance with taking the vaccine within the population. Herd immunity is 60 to 80%. Immunity in the population, I think, to be effective. There have been some different numbers batted around, but I personally think it would take a long time, months, a year maybe, for to get sufficient vaccination with an effective vaccine to have herd immunity.
Mm-hmm. Yeah, I agree totally with Dr. Love. The guesstimates that I've heard about herd immunity is that you're going to need 60 to 70% at least vaccination. And if you think about how quickly you can actually produce the virus, and we have to also convince people to take the vaccination, excuse me. So producing the vaccine, getting it distributed, and convincing people to take it, you're going to need to have 60 to 70% of your population vaccinated and making a good immune response. And so the other thing that we haven't actually talked about at all, what I gave you was a very rosy, good scenario about how your immune system responds and makes antibody. Not all of us, A, make a good immune response, and B, some of the antibodies that we make have the potential to actually facilitate infection and not protect you. So we need to show that the vaccination strategy gives us protective antibodies that produce those antibodies that will block the viral entry into cells, and it is neutralizing antibody. I think that we know from the preliminary data from the vaccination trials that are going on that people are making neutralizing antibodies, so that's good news. But it's going to take a long time. I've heard Tony Fauci say by the end of 2021.
Yeah, well, can I make a little plug for our next seminar in November? We'll be talking about testing and vaccines, and so maybe in a month and a half we'll have a little more information too. So it'll be a good time to update us as we're in the midst of the flu and maybe getting close to testing the vaccine out on more people. So yeah, it will be very interesting. There are a lot of questions coming in. So Pat, you want to ask a question while I go through these and choose some to ask?
Gordon, you were mentioning that the virus seems to disappear from the lungs after a few days. And I was reading that mice and are able to have their dendritic cells traffic some coronaviruses, not necessarily COVID-19, but other coronaviruses. Could this be trafficking it to the lymph nodes or to other organs in the system?
In terms of clearing the virus, I'm not sure that would be sufficient. But and let me restate days, weeks, and the clearing that we've observed, at least on the basis of our immunohistochemical stain, would be patients who have had the disease for a shorter amount of time. And realize that some of our patients have been in ICUs for 2 months. So, and also in these patients who have been in for 2 months, not only are stains negative, but their PCR falls to negative clinically. So I think this disappearance of the virus is kind of backed up on PCR testing clinically, as well as what we're seeing. I think there's an interesting scenario, and we're trying to establish the parameters and look at all of our other tissues. There's an interesting scenario where I think the virus infects and it somehow is disappearing. I think leaving damage to be caused by the cytokine storm or inflammatory processes, as Dr. Osborne went over.
Yeah, can you elaborate on that cytokine storm? That was a question somebody asked as well. So, if Dr. Osborne, you want to share a little bit more about that?
Well, the cytokine storm is something that happens in many, many different infections. But you activate the immune response, and to activate the immune response, you require these cytokines. These cytokines are the good guys. They get the immune response going. However, you can overshoot the good response and make lots and lots and lots of cytokines that will overactivate your system. And these cytokines, that's what causes fever. It's what causes inflammation. These are good effects if done at low levels, bad effects if you have a very high level of cytokines. You get a very high fever, for example. You get vascular dilation. It can be fatal, and I'm sure Dr. Love can speak to that. But normally the immune system is controlled and it shuts off the response, but every now and then it gets out of control and makes a cytokine storm, and what are the good guys turn into the bad guys and can kill patients. Mm-hmm. Would you like to elaborate on that, Dr. Love, about how it kills patients?
I think that I can only elaborate to the extent that what's killing these patients is it's an angiopathy and a coagulation, an aberrant coagulation in the small and large vessels, and that's the end result that we're seeing. How it gets from the virus infection through the cytokine storm to that, I think is the question. We're looking at some other modalities, but we haven't been able to delve into that function.
I'm wondering, in the absence of the trigger for this, the virus, when you lose that virus after a while, what keeps propagating that cytokine storm? Do you know?
Well, that's the question, and I think this is how pathology fits in. I think we need to try to describe what's happening, and then this helps in terms of everyone else then trying to find out what the truth is. So we're just trying to understand the pathologic course of the disease, but we're not at the end point yet.
So my follow-up question may not have a great answer, but seeing as there are a lot of college-age students listening today, the question has come up about long-term effects, even in young people who seem to have mild cases or in some cases asymptomatic. So is there any thinking or idea that there could be long-term effects? I guess it's a two-parter. Young people who do experience symptoms, but then also young people who test positive but don't experience symptoms, that there may be any concern to be had long-term.
I think we have great concerns here. I think that the COVID-19 is going to be a disease that even if you eradicate the new infections, I think it's going to affect medical care for years. Now, I would guess in terms of how people were affected, I would guess that people who are asymptomatic may have a milder long-term effects. But people, what we're dealing with here are plugged vessels. And I think for sure there are brain issues, there are renal issues. And I think that we're going to see continuing pathology, continuing disease by these COVID-19 survivors. And I will give you an example. We've had one autopsy so far of a 34-year-old woman, previously in good health. She had COVID-19 and recovered. And it wasn't a particularly severe problem. She returned about a month later, and she basically died of pulmonary emboli and emboli in the brain. And I think that's we feel that this is a secondary manifestation of COVID-19, and we're looking for more such cases. And I believe that we have a severe disease. This plugging of vessels has to be causing levels of damage elsewhere in the system, and we need to see what's happening in these survivors. But it may not be a simple rosy picture for these poor people.
Mm-hmm.
Now, on the other hand, I don't want to raise alarm, but I mean, it is a concern. And
yeah, no, I think you're right to sort of inform us of the caution, in part because it's so new. So when Dr. Calderer-Moore and I discussed whether or not we should have a theme this year, and are people overhearing about coronavirus and COVID, and yes, we're tired of it, but it's still so new that we have to keep talking about it to be able to learn and do better. So I hate to leave questions unanswered, but I also don't want to keep people here all night. I mean, I do, because I'm enjoying the conversation very much. But I kind of want to get to the big questions that we have as far as what we've learned, where we've succeeded, where we have failed, where we can do better in the future as far as being proactive, maybe mitigating, researching, as Dr. Osborne said, like if this is coming, then where do we need to be as scientists and clinicians to prevent such a thing or be able to stop it sooner in the future? So I'll let you guys choose who wants to start that loaded question and share their thoughts.
Would you like to start that, Dr. Osborne?
Sure. What have we learned? Well, we've learned that we really were extremely rapid, not just us, but the Chinese who came up with the first sequence of the virus, which allowed us to know what we were looking at. But we responded extremely rapidly, and we have started to develop vaccines at a rate that we've never ever seen before. So I'd say that's a huge success. What we also have learned is that we need to have our CDC and FDA better engaged, and we need to have a pandemic task force that's always around and always available. We don't need to create it when we get SARS-4 or SARS-3 or whatever. It needs to be in place now. We need to make sure that NIH, CDC, FDA, they're all available to constantly be thinking about pathogens and new pathogens that might arise.
Very well stated. I think, if I might add, WHO needs to operate freely with full support. We rely on the CDC and other agencies to do the groundwork. And it's very disturbing to see some failures in function.
Dr. Hindmarsh, do you want to add anything?
I think that obviously our response has been very rapid in a lot of ways, but been slow in other ways. And hopefully we're going to learn a lot from this pandemic, because we made some mistakes, but we've had some very good successes. And the next one may be a little bit worse than this. It's hard to believe, but it could be. Now, there was a question over by one of the students about will coronaviruses be the flu of the future? Are we going to have a vaccine every year for the coronavirus? Or are we going to get a vaccine that's both the flu and the coronavirus all together? What do you guys think about that?
Well, first of all, flu and coronavirus are very different viruses. Flu has a mutation rate. It has both antigenic drift and antigenic shift, which cause it to mutate quite a bit, and in fact recombine. Two related viruses can recombine and form a new virus, and so it's a very different beast. And so I don't think that you can equate flu and corona. There is no evidence that this recombination process that takes place in flu is a major driver in coronaviruses. So I don't think we're going to have a new vaccine every year for coronavirus. I think it'll be more like perhaps like polio. It's not the same type of virus, but it will be. One virus one vaccine could immunize you, and maybe you need boosters every year or two. It depends on how long the vaccine is efficacious. If it's efficacious for a couple of years, we maybe only need boosters every 2 or 3 or 4 years.
So that raises another question someone asked about sort of the length of antibodies, right? We hear in the news that it's only a couple of months, but as you mentioned, there's other cells that are involved in an immune response that participate in keeping you immune. So I've always thought we wouldn't be spending all this money on a vaccine if it weren't going to last us for some period of time. I mean, boosters seem reasonable. You have to do that with other vaccines as well. So do you think that it will be effective and will have the long-term immunity that we're hoping for?
We'll see, but I don't think we know yet. We know that antibodies themselves do not hang around for a long period. They have half-lives of months, maybe 6 months at the very most. But memory B cells can live for a long, 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 long time. And so if we create enough memory by vaccination, we should be protected for some period of time.
Mm-hmm. Dr. Calderon-Moore, do you have any last-minute questions before we wrap up? I'll just put you on the spot. Oh, she's muted. She can't talk if she wants to. Can I unmute you?
There we go. I unmuted myself. I do want to thank Dr. Love and Dr. Osborne for joining us today. I was trying to scan through to make sure we didn't miss any questions from the audience, but I think we got most of them.
There are a few that I'm gonna email to our guests, and I think then I can email out to everybody who came. So if certain things didn't get answered, I've made a list, and I'm hoping we can at least get a few more answers. I'm really happy and surprised that there are so many questions. So that makes us feel good about what we've done today, that there is an interest. And sorry, Mary, I cut you off, please.
No, you're good. I was just going to ask both Dr. Love and Dr. Osborne if they had like kind of one parting message, like the last thing that they wanted to kind of summarize to our audience here today.
Well, go ahead, Dr. Osborne.
Now Barbara's muted.
Fund research and get vaccinated. Those are my 2 parting words.
I would like to say in parting words, I'm picking up an idea from different people that this infection only occurs in elderly people who have certain predisposing conditions. And certainly what we found here in New Orleans, we found heavily a racial component among African Americans. We found patients who were overweight, diabetes type 2, certainly. But certainly we know that this disease infects Caucasians. I look at the Italian experience and others. But also there's a wide variety of predisposing conditions, including cancer treatment, immunosuppression of varying kinds. Truly, I would argue with someone who would say that they don't believe the coronavirus is a problem because it will only affect a certain restricted group. I think this is a disease of our time, and it requires every investment that could be made, research and otherwise, to try to control this scourge.
Yeah, and I think that's also a good way for us to. Oh yes, Barbara, please.
I do have one other question. Masks will protect you.
Yes.
Masks are critically important to protect. No, they won't protect you. They will protect you from infecting somebody else. So save a life and wear a mask.
Yes, good advice. Those studies have been really amazing as far as like the hairstylist who had it and saw those customers and didn't infect any of them. It's really amazing how effective they are. In the winter, as Dr. Calderon-Moore mentioned, our series will focus more on healthcare, including healthcare disparities, these different outcomes in patients, the impact this has all had on patient care, how people maybe have had to, I teach bioethics in the winter, and this is like all of that come to life. Who gets access to what in the hospital when, and how decisions like that were being made and who's being impacted. So I think the winter will be an interesting conversation about how patients are being treated and managed during this. So with that, I guess I will close it out and just thank all of you for coming. We had over 130 people attend the seminar, which is really amazing. And we really appreciate you taking the time to join us. Dr. Love and Dr. Osborne, thank you for joining us today and bearing with us as this was our first attempt with this technology. I think it went pretty well, so thank you for your patience and for being a part of it. Maybe one day we'll have you here to visit. Dr. Love, you're not too far away, and Dr. Osborne, I've been trying to get Professor Goldsby here for a while, so it could happen. So thank you again, and thank you, Dr. Hindmarsh, for being a moderator, and Dr. Calderon-Moore for putting this together. Everybody have a good evening. Thank you.
Hope to see you in November.
Thank you.
Thank you.